Welcome to our deep dive, everyone, where we're going to be, like I said, really digging into some really fascinating research on animal behavior in neuroscience.
Awesome.
And we've got a whole stack of sources here,
Yeah.
boat excerpts, scientific articles, all about how scientists use animal behavior to model human conditions and drug effects.
Yeah, it's a really interesting way to think about how we can take what we see on the outside and kind of peer into the brain a little bit.
Yeah, it's like getting a window into this, like, black box in a way.
Exactly.
And one of the things that I thought was really interesting just in starting to go through all these different sources, just
Mhm.
like how important it is to choose the right research subject,
Oh, absolutely.
right? It's not just like picking any mouse off the shelf.
Right, like a mouse is a mouse is a mouse.
Right.
But as we've seen, that's not really the case. Different species, even different strains within a species, can react in totally different ways to the same treatments.
Yeah. And there's this fascinating example in one of the articles about hippocampectomy. And just to remind everyone, that's where they surgically remove part of the hippocampus.
Right.
And what they found is that Sprague dolly rats
Okay.
actually become more active after the surgery.
Wow. So that's counterintuitive.
Yeah.
You'd think if you're taking out part of the brain, it would reduce activity, but not
Well,
necessarily.
but then you look at Fisher rats, which are generally less active to begin with,
Uh-huh.
they become even less active.
So it's really highlighting those baseline differences that we need to consider.
Right, and how those can actually influence the results of your research.
Makes
So it's
sense.
like you were saying, a mouse is not a mouse is not a mouse.
Exactly.
But then it makes you wonder, when we're thinking about all this research and how much of it is done on animals,
Yeah.
like how much can we really generalize, from one species to another, especially when we're thinking about really complex human conditions?
Yeah, that's the big question, right?
Right.
How much can we actually take these findings and extrapolate them to humans?
And even within, a species,
Yeah.
there's so much variation.
Oh, absolutely.
Like even the history of the albino lab rat.
Right. The classic.
Yeah, the classic research animal.
Right.
It's kind of like a mystery, like where it actually came from.
Yeah, there's some debate about its true origin.
Right, like it's potentially descended from wild rats that were captured in the US.
Uh-huh.
But then there might also be some European influences mixed in there.
Yeah, we don't know the full story.
Yeah, it's so interesting to think about all these like hidden genetic factors that we might not even be aware of.
Right. It's like this hidden layer
Yeah.
of complexity to the
Like-
research.
And how that could be influencing the results without us even knowing.
Absolutely. And it's not just rats. Either primates, which are often used in these more complex behavioral studies, have their own considerations.
Yeah, like all those rhesus and cynomolgus macaques and apella monkeys that our sources mentioned.
Right. Each of those species has its own quirks, its own behavioral characteristics.
So what are some of the key differences between those different types of monkeys?
Well, for example, apella monkeys are known for their incredible dexterity.
Okay.
They're really good with their hands. They can manipulate objects in a really precise way.
So if you were studying something that involved fine motor skills,
They'd be a great choice.
they would be a great choice.
Exactly.
Okay, so it's all about really thinking about the question you're asking and matching that to the right animal model.
Absolutely. The choice of species is crucial.
So we've talked a bit about rats and primates broadly.
Yeah.
But let's kind of dive into some of the specific behavioral paradigms that researchers use with mice,
Okay. Sounds good.
because we have a lot of mouse research in here as well.
Yeah. Mice are incredibly valuable in neuroscience research.
And we were talking a bit before we started recording about how, even if you're studying something like memory or cognition,
Right.
you still need to have this understanding of the animal's basic ability to sense and respond to things.
Absolutely. You don't want to misinterpret what looks like a cognitive deficit for something that's actually a sensory or motor problem.
So how do you actually go about assessing that in mice?
Well, one common way is through the acoustic startle response and prepulse inhibition, or PPI for short.
Okay, so break that down for me.
So for the acoustic startle response, you're basically just exposing the mouse to a sudden loud noise and seeing how much it jumps.
Okay, so like a little jump test.
Exactly.
Yeah.
It gives you a baseline measure of how sensitive their nervous system is to unexpected stimuli.
So like a really jumpy mouse might have a more sensitive nervous system.
You could interpret it that way.
Okay, interesting. But then you said there's this other thing called prepulse inhibition.
Right. That's where things get even more interesting.
Okay, so tell me about that.
So with PPI, you're introducing a quieter pre-pulse sound before the loud noise.
Okay, and what does that tell us?
Well, a normal mouse will be a little startled by the pre-pulse, and as a result, their startle response to the louder noise will be less intense.
Okay, so they're kind of prepared.
Exactly.
They're able to filter out that irrelevant information.
But if they have some kind of impairment in their sensory processing or inhibitory mechanisms,
Right.
that pre-pulse won't really do anything.
They'll still jump just as high at the loud noise.
So it's like their internal volume control is broken.
That's a great analogy.
I like that.
So, one interesting thing about this is that mice actually need more sensitive equipment for measuring their startle reflex than rats do.
Oh, that's surprising. Why is that?
Well, it's simply because of their size. Mice are so much smaller than rats that their startle movements are less forceful.
Yeah.
So you need more precise equipment to pick up on those subtle responses.
So it's not just about like the mouse jumping, it's about like all the technology and the nuance
Exactly. There are all these layers of complexity.
that goes into measuring these things.
Right.
But why should we care about a mouse's startle response?
Well, studies have shown that scopolamine, a drug that affects the cholinergic system, disrupts PPI in mice.
Okay, but how does that relate to humans? Well, that's the fascinating part. Deficits in PPI are also observed in human disorders, like schizophrenia.
Oh, wow.
So these seemingly simple tests in mice can actually offer clues about potential mechanisms underlying complex human conditions.
So we're not just talking about mice jumping here. We're talking about potentially unlocking new avenues for understanding and treating schizophrenia.
Exactly.
It's incredible.
And there are a whole bunch of other methods for assessing motor function and exploration in mice, like the rotorod,
Okay.
which tests their balance and coordination,
Right.
and the beam balance, or walking task, where they have to navigate a narrow beam.
It sounds like those would be really sensitive to things like the mouse's anxiety level.
Oh, absolutely.
Or even like the time of day you're testing them.
Right. A stressed out mouse in the evening probably isn't going to perform as well as a relaxed one in the morning.
Right. It's all about context.
Exactly. And even things like the genetic strain of the mouse can have an effect. Researchers have to be incredibly meticulous about controlling for all of these factors.
So many variables at play.
There-
It's not just about observing a behavior. It's about understanding like the whole context in which that behavior is occurring.
Right. And that attention to detail is what allows researchers to uncover those subtle but crucial insights.
Absolutely.
Oh.
Okay, let's talk about another really interesting paradigm.
Good.
Conditioned taste aversion, or CTA.
Ah, yes.
Yeah.
CTA, classic example of how powerful simple forms of learning can be.
Okay, so how does it work?
Essentially, you pair a novel taste, something the animal hasn't encountered before, with something that makes them sick.
So they learn to avoid that taste in the future
Exactly. Even if it wasn't the taste itself that made them ill,
because they've made that association.
right?
Okay, so it seems like basic learning, but you said it's a really powerful tool in neuroscience.
It is. It's used in drug discrimination learning, which we'll get to a bit later.
Okay.
But it also allows researchers to study the subjective effects of drugs, which is something you can't really do with traditional methods.
Can you give me an example?
Sure. One of our articles talks about cyclophosphamide, a drug that can cause nausea. By pairing it with a specific taste, researchers can measure the strength and duration of the taste aversion that develops.
So a mouse that really strongly avoids that taste suggests that the drug produced a really powerful and lasting effect.
Precisely. And what's really cool is that the magnitude and longevity of the CTA can change depending on things like the dosage or how close in time the taste and the drug are paired.
So it's like they're really fine-tuning their understanding of how those variables influence the effects.
Exactly.
And it all comes down to observing like a simple behavior avoidance.
It does. It's all about the details.
Wow. Okay, so I think we've covered a lot of ground already.
We have.
But I'm really excited to move on to another paradigm.
Okay.
Drug discrimination learning, or DDL.
All right, let's dive into the mind of a mouse.
Yes, let's do it. So what is DDL, and how does it give us a glimpse into what a drug feels like for an animal?
Well, imagine you're training a rat to tell the difference between a stimulant like amphetamine and saline.
Okay.
In DDL, they learn to respond differently depending on which one they get.
So they learn if they get amphetamine, they should press one lever. If they get saline, they press a different lever.
You got it. And once they've mastered that discrimination, researchers can then test whether other drugs produce similar subjective effects.
So if the rat gets a new drug and presses the amphetamine lever, it suggests that that new drug produces a similar internal state to amphetamine.
Exactly. We're getting a glimpse into their subjective experience, even though they can't tell us how they're feeling.
Wow. It's amazing what scientists can do,
It is.
but it's so clever.
It is.
And what's even more remarkable is that drugs that produce similar effects in humans often show this generalization in animal DDL studies.
Right. So if a drug makes both rats and humans feel euphoric, The rats are more likely to generalize that drug to a known euphoric drug in the test.
So there's like this common underlying experience.
Exactly, across species.
Which is fascinating.
And that's what makes these findings so translationally relevant.
Because it suggests that what we learn in animals
That's right.
could actually have real implications for humans. Okay, so we've talked about sensorimotor processes. We've talked about taste aversion.
Yeah.
We've talked about drug discrimination.
We have.
What other fascinating paradigms await us?
Well, let's move on to a paradigm that explores the realm of motivation and reward.
Okay.
Conditioned place preference, or CPP.
Okay. I'm sensing a theme here.
Lots of conditioning.
Lots of conditioning.
Yeah.
But how does CPP work?
So think of it this way. You expose an animal to two distinct environments, like chambers, with different visual or textural cues. And then you pair one of those environments with a drug or something that's thought to be rewarding.
Okay.
And if the drug is indeed rewarding, the animal will start to prefer spending time in the environment that was paired with it.
So they're seeking out that good feeling.
Exactly. They're making that association between the place and the positive feeling.
And is there like a flip side to this, like a conditioned place aversion?
There is CPA, where the animal learns to avoid a location that's been associated with an aversive experience.
So it's like a bad taste with a bad place.
Exactly.
Okay, so both of those sound really useful for studying like the motivational properties of drugs and how animals learn to approach or avoid certain stimuli.
Right. It's all about understanding what drives behavior.
Exactly. But as with any research method, I imagine there are some like methodological things to keep in mind.
Oh, absolutely.
Right, like our sources mention that it's really important to test animals in a non-drugged state in CPP and CPA.
Right. You don't want their behavior to be influenced by the drug's direct effects.
So why is that so important?
Well, if the animal is tested while they're still under the influence of the drug, their behavior might be influenced by just the drug itself and not their learned association with the environment.
Right, so you want to make sure that it's the memory of the experience.
Exactly.
Not the drug itself.
It's about teasing apart those factors.
Right, that makes a lot of sense. But this kind of reminds me of something we talked about earlier, state-dependent learning.
Oh, right.
Could that be a confound in these studies?
Absolutely. Remember, state-dependent learning is when what an animal learns in a specific drug state is more easily retrieved when they're back in that same state.
Right, so if a mouse learns something while it's on a stimulant, it might struggle to remember it when it's not on the stimulant.
Exactly. Their memories are tied to their internal state.
Wow, so researchers really need to be mindful of that
They do. It's a crucial consideration.
and design their studies accordingly.
Right.
Maybe include test sessions in both drugged and non-drugged states to really get a clear picture.
Exactly. It's all about careful design.
Okay, so much to consider.
There is.
I think we have time to squeeze in one more paradigm before we wrap up this first part.
Okay, let's do it.
Let's talk about primate models of drug self-administration.
All right, now we're getting to some really complex and unfortunately a bit closer to home territory.
Why do you say, unfortunately, what makes primate models so valuable in this context?
Well, primates, particularly those closely related to humans, can exhibit patterns of drug use that are disturbingly similar to what we see in people.
You mean they can become addicted?
Sadly, yes. And that makes them a powerful, albeit somewhat unsettling tool for studying the neurobiological mechanisms underlying addiction.
So how do researchers actually study this in primates?
There are a variety of methods,
Mm-hmm.
but a common one is to train the animals to press a lever to receive an intravenous injection of a drug like cocaine or heroin.
And they'll actually work for the drug.
They'll press that lever over and over again,
Yeah.
even if it means expending a lot of effort.
It's really highlighting how strong that reinforcement is.
It is. Those drugs have powerful reinforcing properties.
And to really understand those properties, researchers can manipulate the schedule of drug delivery.
Exactly.
Okay, so what do you mean by schedules of drug delivery?
Well, you have things like fixed ratio schedules, where the animal has to press the lever a set number of times to get a dose.
Okay.
Like 10 presses equals 1 dose.
So they have to work for it.
Exactly.
What other schedules are there?
You also have fixed interval schedules where a certain amount of time has to pass before a lever press will deliver a dose.
Okay, so it's not just about how much they work, but when they work.
Exactly.
Interesting.
And there are even more complex schedules like second-order schedules and progressive ratio schedules that help researchers uncover different aspects of drug reinforcement.
It sounds like a whole world of research in itself.
It is. There's so much we can learn from these different schedules.
So what are some of the key takeaways from these studies?
Well, one crucial finding is that different drugs have different reinforcing potencies.
Okay, so some drugs are just more addictive than others.
Exactly.
Can they actually quantify how much more addictive one drug is compared to another?
They can. Our sources describe a study where cocaine was found to be 10 times more potent than procaine in a progressive ratio procedure.
Wow.
So the animals were willing to work much harder for cocaine.
That's a huge difference.
It is. It really highlights the power of these studies.
It's like they're going beyond just observing whether an animal will self-administer a drug. They're getting at the underlying mechanisms.
Exactly. It's about uncovering those subtle ways in which drugs exert their influence.
So fascinating and a bit disturbing at the same time.
It is, but understanding the neural basis of addiction is crucial.
Absolutely, if we want to develop effective treatments.
Exactly.
Well, I think we've covered an incredible amount of ground in this first part of our deep dive.
I think so too.
We've gone from mice jumping at loud noises to primates making really complex decisions about drug rewards.
It's been quite a journey.
It has been, but there's so much more to explore, so be sure to join us for part 2, where we'll delve into the mysteries of memory and how researchers study something so complex and elusive in animals.
Looking forward to it. We really did cover a lot in part 1.
We did. It was a lot of information.
Like from the very basics of just like, can a mouse even sense and respond to things?
Right, right.
All the way to like primates making decisions about whether or not they want to take a drug.
Right, and how addictive those drugs might be.
Exactly, and how we can measure that in the lab.
But, there's this one big area that we haven't talked about yet.
Oh, yeah.
And that's memory.
Ah, memory, the big one.
It's like the ultimate puzzle of the brain, isn't it?
It really is. Like, how do we form these memories? How are they stored? Why do they sometimes fade? Why do they change?
It's such a mystery.
It is. And researchers have been trying to answer these questions for decades using all sorts of clever methods with animals.
Right, because we can't exactly, crack open a human brain.
Not ethically, anyway.
Right, exactly.
So we have to rely on these animal models to give us clues.
So what are some of the ways that scientists actually study memory in animals?
Well, one of the classic methods, and you might be surprised to hear this, but it's considered a relatively simple method, is inhibitory avoidance.
Okay, inhibitory avoidance simple, you say?
Relatively speaking, of course, in the grand scheme of memory research.
Okay, I'm intrigued. Paint me a picture.
Okay, so imagine a two-compartment chamber. One side is brightly lit. The other side is dark. Rodents naturally prefer the darkness.
Okay, makes sense.
So when they're given the chance, they'll scurry right into that dark side.
Right, to feel safe and secure.
Exactly. But here's the catch. When they enter the dark side, they receive a mild electric shock to their feet.
Oh, ouch.
Yeah, not pleasant, but it's a very mild shock. Don't worry.
Okay, but I bet they learn that lesson pretty quickly.
They do, and that's the whole point. The next time they're placed in that chamber, they'll hesitate or even completely avoid going into the dark side.
Even though they want to.
Exactly. Even though their natural instinct is to seek out the darkness, the memory of that shock makes them inhibit that behavior.
So the longer they hesitate, the stronger their memory.
You got it. It's a simple but really elegant way to measure memory retention.
Wow, I like that. Simple but elegant.
And it's not just for basic memory research either. This paradigm has been used to study how drugs affect memory,
Okay.
the role of different brain regions in memory, even age-related memory decline.
So many applications.
It's a very versatile tool.
It sounds like it, but I imagine there are some caveats to consider when you're interpreting the results.
Oh, absolutely. Like with any behavioral paradigm, you have to be careful about over-interpreting things.
Right, because animal behavior can be complex. There could be other things going on.
Exactly, and our sources point out some important things to keep in mind with inhibitory avoidance, for example. Just because an animal avoids the dark side doesn't necessarily mean they have a good memory.
Okay, that's interesting. What else could be happening?
Well, they could be more anxious in general.
Mhm.
Maybe they're just hesitant to move around a lot, not specifically because they remember the shock.
Right, so it's not just about the avoidance itself. It's about understanding the animal's overall behavior and looking for other potential explanations.
Precisely, and that's where good experimental design comes in. Researchers might use additional tests or carefully observe the animal's behavior in other contexts to rule out those confounding factors.
So it's all connected.
Behavior is complex and multifaceted.
Well, let's move on to another memory paradigm. This one I'm particularly excited about because it just sounds so cool. The radial arm maze, or RAM.
Yeah, yes, the RAM, a
That's-
classic for a reason.
Okay, tell me everything. I'm picturing a little rodent scientist, with a little notebook and a tiny pencil mapping out the maze.
Well, picture this, a central platform with multiple arms radiating outward like spokes on a wheel or a starburst. And at the end of each arm, there's a little tasty treat waiting to be discovered.
Oh, so the rodent's job is to find all the treats.
Exactly. It sounds like a pretty fun job, right?
It does, but I'm guessing it's not quite that straightforward.
You're right. Here's where the research magic happens by changing the rules of the game. Scientists can use the RAM to study different types of memory.
Okay, now you've really piqued my interest. What kind of rules are we talking about?
One common procedure is called the wind shift task. In this task,
Hmm.
the rodent has to remember which arms they've already visited and avoid going back to them, even if those arms still have treats.
So they can't just rely on their sense of smell or just like a simple strategy of go where the food is?
Exactly. They have to actively keep track of where they've been and update their mental map of the maze as they go.
So they're really using their working memory.
Exactly, that ability to hold information in mind and use it to guide their actions.
It's like when you're trying to remember a grocery list without writing it down.
A perfect analogy. You have to keep track of what you've already put in the cart so
Yeah.
you don't end up with 5 cartons of milk.
Exactly. And researchers can track all sorts of things in this task, like how many errors the rodent makes, how long it takes them to find all the treats, even the specific pattern of their arm choices.
Right, they can really get a detailed picture of the rodent's spatial working memory
Uh-huh.
and how it's affected by different factors.
So they might compare how the animal performs under different conditions, like after being exposed to stress or given a drug that might impair memory.
Exactly. It allows them to tease apart the nuances of how memory works in different situations.
Fascinating. Now, you mentioned another procedure earlier, the delayed non-match to sample task, or DNMTS.
Ah, yes, DNMTS, another classic. This one taps into a different kind of memory recognition memory.
Okay, remind me, what's recognition memory again?
It's our ability to remember whether we've encountered something before. Like, have I seen this person before? Have I been to this place before?
Okay, so it's less about spatial navigation and more about recognizing familiar things.
Exactly. In DNMTS, the rodent first learns that a particular arm is always baited with a treat.
Okay.
Then after a delay, they're given a choice between that familiar arm and a new, unvisited arm.
And they have to remember which one had the treat before and choose the different arm to get the reward?
You got it, it's like a game of what's different for rodents. And the longer the delay, the harder it is for them to remember.
So it's really testing the limits of their memory capacity and how long they can hold on to that information.
Precisely. And like the windshift task, DNMTS, has been used to study a wide range of things, from the effects of brain lesions to the potential of new drugs for memory disorders.
It's amazing how much we can learn about these really complex processes just by watching rodents run around in a maze.
It is it highlights the power of behavioral analysis and how much we can infer about what's happening in the brain just by observing the animal's actions.
Well, my brain is already feeling full from all this memory talk.
I know there's a lot to unpack, but we're not done yet.
Oh, there's more.
There's always more when it comes to the brain. In part 3, we'll dive even deeper into the world of memory, looking at some even more complex procedures and some of the theoretical considerations that guide this fascinating field of research.
Okay, I can't wait to join us for part 3 as we continue to unravel the mysteries of the brain, one behavioral paradigm at a time. Welcome back to our deep dive. I feel like we could talk about memory forever.
Oh, easily. It's such a rich and fascinating topic.
It really is, but I have a feeling we've only just scratched the surface.
Oh, yeah. We've covered some of the foundational paradigms, but researchers have developed even more intricate and complex procedures to really probe those fine details of memory in animals.
Okay, so lay it on me. What are some of these advanced memory tests, and what can they tell us about how memory works?
Well, one that stands out is the concurrent spatial discrimination task. This one really takes things to a whole new level of complexity.
Concurrent spatial discrimination, that sounds intense. How does it differ from those RAM tasks we were talking about before?
So instead of just remembering individual arms they visited in this task, the rodent has to learn a specific rule about pairs of arms in the maze.
Pairs of arms. Okay, you've got my attention.
So picture 2 arms right next to each other. Let's call them arm A and arm B. In this task, one arm is always going to have a treat, while the other is never baited.
So it's not about remembering which specific arms they've been to, but more about learning a consistent pattern within the maze.
The rodent has to figure out that, for example, arm A is always the good one, while arm B is always a dead end no matter what.
So they're not just forming a memory of individual locations, they're actually learning a spatial rule.
Exactly. It's like cracking a code, figuring out the underlying pattern, rather than just memorizing a list of places.
And this is particularly useful for studying what kind of memory.
This is great for looking at reference memory.
Okay, remind me, what's reference memory again?
Think of it like our long-term knowledge of the world, the stuff we just know, our home address, our friends' names. Those are all examples of reference memories. They're stable and enduring.
So in this concurrent spatial discrimination task, the rodent is basically building a reference memory about which arm always has the treat. They're not just remembering a specific event, they're forming this long-term understanding of the maze's layout.
Precisely. And this allows researchers to study what brain regions and neurochemicals are involved in forming and retrieving these types of long-term spatial memories.
It's incredible how much we can learn about these fundamental brain processes just by observing these rodents in a maze.
It really is, and the creativity of the researchers who design these tasks is just amazing.
Absolutely. Are there any other advanced procedures that come to mind?
Another really interesting one is the delayed non-matching to place task, or DNMTP. It builds on the delayed non-match to sample task we talked about before, but with a spatial twist.
Okay, I'm trying to remember what was delayed non-match to sample all about again.
Right, so in that task, the rodent had to remember a specific object or cue and then choose the different object when given two options.
Okay, yeah, testing their recognition memory.
Exactly. But with DNMTP, instead of remembering an object, they have to remember a location, specifically the location where they previously found a reward.
So it's a spatial memory test, but with that delayed component to make it even more challenging.
Exactly. And it involves this clever combination of forced choices and delayed choices.
Okay, break it down for me. How does this forced choice element work?
So in the first part of the trial, the rodent is directed into one specific arm of the maze where they find a treat. Let's say it's arm A.
Okay, so they don't really have a choice at that point.
Right. Then they're directed into a different arm, arm B, which doesn't have a treat, just to ensure they experience both arms before the real test.
So it's like setting the stage, making sure they're familiar with both locations.
Exactly. Then, after a delay, the rodent is placed back in the center of the maze and allowed to choose between arm A and arm B.
And to get the reward, they have to choose the arm that didn't have the treat earlier.
You got it. They have to remember which arm was baited even though they've experienced both arms since then.
So they have to inhibit that natural tendency to return to the place where they most recently found food.
Exactly. It's a test of not only their spatial memory, but also their inhibitory control.
And by changing the length of the delay, researchers can really see how long that special information is held in memory.
Right. They're essentially probing the limits of spatial working memory.
Wow, these tasks are so clever. I'm in awe of the researchers who come up with this stuff.
Me too. It's a really testament to human ingenuity and our desire to understand the brain.
Absolutely. But you know, it's not just about designing clever experiments. Our sources also talked about some of the theoretical and practical considerations that have really shaped the field of memory research.
Right. It's important to have that broader context.
Yeah. So how has our understanding of memory evolved over time?
Well, early research was really dominated by what were called SR theories, stimulus response.
Okay, stimulus response.
The idea was that learning and memory were all about forming simple associations between a stimulus and a response.
Kind of like input, output.
Exactly, like we learned that fire is hot so we don't touch it.
Right, a pretty basic survival mechanism.
But as researchers started to dig deeper, they realized that memory was far more complex than just linking inputs and outputs.
Because we can learn and remember things without always making an outward response.
Exactly, and that led to the understanding that there are actually multiple memory systems in the brain, each with its own unique properties and functions.
So we've touched on a few different types already: working memory reference, memory recognition, memory. What other memory systems are there?
Well, there's procedural memory, which is our memory for skills and habits like riding a bike or playing the piano. And then there's episodic memory, which is our memory for specific events and experiences.
So the different types of memory tests we've been discussing are actually tapping into these different brain systems, each with its own neural underpinnings.
Exactly, and that realization has really revolutionized the field of memory research. It's not enough to just use one type of task. We need a whole toolbox of tasks to really explore these different systems and how they interact.
So it's not just about the apparatus they're using like a maze or a Skinner box. It's about the cognitive demands of the task itself.
That's a key point. Researchers have to think carefully about what the animal needs to learn and remember to be successful in the task. Are they forming simple associations, or are they building a more complex cognitive map?
It's all about choosing the right tool for the job and understanding those nuances.
Precisely, and this has led to the development of increasingly sophisticated and sensitive behavioral paradigms, constantly pushing the boundaries of what we can learn about memory.
It's really inspiring to see how far this field has come and how much we've learned from studying animal behavior.
I agree. It's a testament to the power of scientific inquiry and our relentless curiosity about the brain.
Well, I think it's safe to say that our brains are officially full after this deep dive.
I think mine is overflowing.
We've covered so much ground from those basic reflexes to the complexities of memory and everything in between.
We've seen how animal behavior can give us these incredible insights into the inner workings of the brain.
Insights that can ultimately lead to new treatments for human diseases and a deeper understanding of ourselves.
And that's really what it's all about, using this knowledge to improve human health and well-being.
Absolutely. So as we wrap up this episode, we want to thank you for joining us on this incredible journey, and we encourage you to continue exploring the fascinating world of neuroscience.
Keep asking those questions, keep learning, and never stop being curious about the brain.
Until next time.